quindi tu farai finta di fare una riunione. Questo è il concetto.
Good afternoon, everyone, and welcome to today's IBMA webinar on peptides and proteins as plant protection products, technologies, and IBMA actions. I'm delighted to have you all here with us. Today, we have an exceptional registration and also participation. Thank you for everyone being here. Peptides and proteins are increasingly emerging as innovative solutions in plant protection, offering new ways to support sustainable agriculture. However, alongside the scientific promise, there are regulatory considerations that must be addressed to ensure their successful authorization and use at both EU and international level. Since 2020, IBMA have been working on what we would like to call our peptide journey. Step by step, this journey has aimed to build clarity, guidance, and a solid foundation for the development and approval of peptide and protein-based products as biocontrol solutions. Today, we are very fortunate to be joined by two distinguished experts who will share their scientific insights and perspective on this evolving field. First, I would like to welcome Professor Emilio Montesinos, Professor of Plant Pathology and Microbiology at the University of Girona, Institute of Food and Agricultural Technology. And his presentation will explore functional peptides for plant disease control, sources, developments, and the challenges. Then we'll follow Dr. Andrea Chini, Senior Scientist at the National Center for Biotechnology, Madrid, holding a PhD in plant molecular biology from the University of Edinburgh. Dr. Chini will take us on a deep dive on emerging technologies and opportunities for natural and natural-like molecule in plant protection. Their expertise, combined with IBMA's regulatory action, will provide us with a comprehensive overview of both the scientific opportunities and the challenges ahead for peptides and proteins as plant protection products. But before we begin, just a quick note on a few housekeeping points to help the webinar flow smoothly. First, you are welcome to submit your questions in the chat. We will collect them and bring them during the Q&A session that follow the three presentations. To ensure a smooth flow, we kindly ask you to hold verbal intervention until the Q&A session at the end of the presentations. And finally, please note that the session is being recorded and a replay will be made available to IBM members as usual. With that, let me thank you once again for joining and hand over to Jennifer Lewis, who will describe us the IBM actions and strategy on proteins and peptides in more details. Thank you very much. And Jennifer, the floor is yours.
Thank you, Jeremy, and thank you to all the members attending, and welcome. I add my welcome to that of Jeremy's to talk you through the peptide journey, as we call it, that IBMA has been on and continues to be on. So I wanted to just reflect a little bit on this journey, so where we started, where we've got to, and where we're going to. Because we should recognise that within all this work that we're doing and that our members have done, there is a lot of work going on outside the organisation and in government organisations, intergovernmental organisations, advisors, academics. And so it's very important that we have that holistic view on everything that's happening. So we first started in 2020, as Jeremy mentioned. We actually did a scoping committee just before COVID on peptides and proteins, and included all identical peptides and proteins into the IBMA definition. Then we got to 2023, where there was an increasing amount of discussion, both among our members and outside in the commission, and it was raised as a point for discussion in the biopesticide working group. On the back of that, the request both of the commission and some of our members, we set up a scoping committee to look at peptides and proteins and to see how those could be reviewed and what they were in terms of fit to the IBMA definition. During this process, which took about a year and a half, over 18 months, in fact, we had a meeting at OECD, which included a case study, a joint case study with CropLife, where we looked at identical peptides and the type of things that would need to be considered in approving such molecules. It was then agreed that we would then have a seminar in 2025 to include regulatory considerations on peptides. So this was a result of quite some discussion between IBMA and OECD, because as IBMA, we felt it was very important that the intergovernmental organisations started to consider this as well. And they were indeed very keen to do that. Towards the end of 24, we concluded our IBMA scoping committee, and the peptides under certain conditions were included in the IBMA biocontrol definition. And the next step then was to actually move to a working party that could start to prepare for the subsequent OECD discussions, as well as look at what the options are for bundling peptides and antibodies in a similar way to senior chemicals. If I can have the next slide, please. So now really what we see is the discussion being in OECD, in the European Commission, in the member states. It's now an important discussion. Why? Because these molecules are increasingly important in plant protection products. So at the OECD seminar in February of 25, there was a seminar regarding peptides, protein and peptides, and various academics, and some of them present here, thank you, gentlemen, and other experts presented on the matter. What became clear was that it would be relevant to have further thought as to what and how these molecules should be regulated. And indeed, it was agreed that we should consider a so-called considerations document, which OECD is looking at what that should be, and has asked IBMA to prepare some terms of reference, which duly is being done by the Working Party on Proteins and Peptides. This will then have a further discussion at ABIM, so at our annual biocontrol industry meeting in October in Basel. There will be a workshop which will further discuss what and how to regulate proteins and peptides, and all companies are welcome. There will be presentations there from regulators and from OECD and from IBMA, and we will welcome the discussion with all companies at that point. In 2025 to 2026, after we've prepared this considerations document for further discussion in OECD, then there will be further work, probably looking at how we can enlarge our activity in proteins and peptides in order to meet the needs of the farmers as they go forward. Perhaps in the next slide, we can look a little bit at the companies that we're currently working with. So you see in sort of the pinky color, the companies that are currently working on peptides within IBMA and involved in our discussions, or have licensed someone who's involved with IBMA. We're also in discussion with several of the other companies, again, to maximize the contribution to these consideration document that is being prepared, or the terms of reference for it. Because what we would like to achieve is that we have the best possible information available, the widest possible consideration that goes into what will then be discussed at OECD in February of next year. So to summarize, we've been on a journey. We're still on the journey. We're really pleased to have all these members actually working with us on this matter, and believe that it's an extremely important topic for crop protection going forward. And really, this discussion is now happening and will be very relevant overall in the biological definition discussion as we go forward into the simplification procedure. So, without further ado, thank you, Jeremy. I will pass back to you. Thank you. I think you might be on mute, Jeremy.
Sorry. Thank you very much, Jennifer. Now we will give the floor to Professor Emilio Montesinos. Professor Montesinos has more than 30 years of experience in plant pathology, bridging research and practical application in biocontrol, and particularly peptides. As I mentioned earlier, he will, in his presentation, he will explore the functional peptides for plant disease control, dealing with the sources, development, and challenges in the matter. So without further ado, the floor is yours, Emilio. Thank you very much.
Thank you very much, Jeremy. I hope everybody is seeing my first slide. Can you hear me also?
Yes. Yes.
Okay.
Slide
okay.
on and hear well.
So I will start. First of all, I would like to thank IBMA for inviting me to this webinar. It's very respectable number of attendants, so I'm a little bit nervous. But well, I will focus on my specialty, which is plant disease control. I will not talk about insect control, which is similar but a different way. We learn from nature about functional peptides because we can find these compounds in animals and plants, and also in microorganisms. As a consensus, we consider peptides polypeptides of less than 50 amino acid residues, but also very small proteins. In animals and plants, the function is generally the first barrier of innate immune defense. In mammals, for example, but also in insects and in plants. The world of microorganisms is very rich in production of peptides, especially antimicrobial peptides. And in most cases, they are the responsibles of the antibiosis. We can divide in many ways the world of peptides, but I prefer a system which is based on the way of natural synthesis in the living cells, which is the ribosomal peptide production, which is protein synthesis, the conventional one, and there are many examples of them, but also, probably more important in our field are the non-ribosomal peptide production, which is produced by an enzymatic pathway, very complex generally, and this is typically produced by microorganisms. Up to now, we know more than 20,000 different types of compounds. From that, more than 7,000 are antimicrobial peptides, and most of them are non-ribosomally produced peptides. You can find information on that in several databases like AlphaFold, PerFold, UniProt. Probably Dr. Chini will talk more in detail about that. Attending on microorganisms, and I know that many associates to the IBMA know very well the world of microorganisms, especially the world of biocontrol agents, is very rich in strains producing especially antimicrobial peptides. For example, in our research group, we have developed bacillus strains, for example, these two strains, producing cyclolipopeptides and bacillicin. This is a patent of a company. Lactic acid bacteria are very interesting because produce bacteriocins. For example, the species Lactobacillus Lactoplantobacillus plantarum produce plantaricins. The Pseudomonas, which is very popular in the world of biocontrol, especially Pseudomonas fluorescens, is producing cyclolipopeptides also. Even the Panthoeas, that are Enterobacter or Erwinia in the past, is the synonymous name. Panthoea glomerans produces pantocin A. Delphtia is very interesting because it's a component of several insects and also plants, producing delphibactin. And Streptomyces produces a huge amount of different diverse antibiotics. So these are 6 examples of where we can find antimicrobial peptides. But in many cases, these peptides are produced in low concentrations or are toxic or have a profile that is not biologically interesting for us. So what we do is to develop analogs based on the natural ones with the objective to improve their properties, or even to design new peptides invented on the basis of molecular design. For example, this is the platform we use together with a peptide chemistry group at my university, the LIPSO. So departing from the natural ones and using molecular design, especially based on a structure, function, and target-oriented approach, which is a very complex system. Probably Dr. Kinney will talk about that. We develop theoretically a list of compounds, a library that is synthesized by solid phase synthesis, and then these compounds are tested, usually using a high throughput screening for antimicrobial activity against different plant pathogens, and also for defense elicitation. For example, in the past we have been using tobacco cell cultures. And then the lead peptides that fit the best condition were submitted to a second round of molecular design. And after a few rounds, we find a few compounds that are really interesting, but we continue the screening because usually it's important, for example, to see what is the hemolytic activity, the protease stability, and some preliminary toxicity testing. Of course, also proof of concept, which is efficacy efficacy testing in vitro and also in planta. And those improved peptides are the ones that selected into the pipeline. Several mechanisms of action have been described among the antimicrobial peptides. You can see here a review I did in Annual Review of Phytopathology 2 years ago, but the most popular mechanism is the lytic activity. So they destroy the cells of prokaryotes or eukaryotes simply by producing distortion in the membrane and making holes and pores, and then the cell is really inactivated. But there are also peptides that affect internal processes, for example, protein synthesis. Other peptides could interact with external structures. For example, we have peptides that interact with lipopolysaccharides. In this way, they produce an effect of inactivation of the cell. Other peptides are antibiofilm peptides, and you know that many of the pathogens, plant pathogens, especially bacterial plant pathogens, mediate pathogenesis by the production of biofilm. So this is important. Other peptides produce interference with spore germination, which is very important, obviously, in the infection process of several fungi. There are also peptides that are nematicidal. Even there are a few descriptions of peptides that are antiviral. So you see that there are several mechanisms of direct action, but this is not all about the mechanism, because one of the most interesting mechanisms involving peptides is an indirect action on the plant by elicitation of the defense response or a stress relief response. So probably you know that there are many elicitors, including microorganisms, insects, mechanical lesions, chemical compounds, drought, thermal and saline stress, and so on. But peptides also, several peptides can induce defense mechanisms of the plant. Probably also you know that these elicitors mediate by means of messengers that are very well known: jasmonic acid, ethylene, salicylic, abscisic, auxins, gibberellins, peptides, again, as messengers, and also in the last years, microRNAs and nitrous oxide. So these messengers move up and down into the plant and produce the one response that we know as systemic response into the plant. Into the plant, there is an effect that finally produces a response. The most known response is the reactive oxygen species burst, which is an oxidative burst to defend themselves against the pathogens. There are proteins, PR proteins, that are very well known also, phenolic compounds of different types. So important that some peptides can induce this response into the plant. I want to give you an example of an analog peptide, which is a chimeric or engineered peptide we have developed in my lab, which is based on fragments of secropin, melittin, and magainin, which are three peptides from insect origin. And we have improved these sequences, and we have added sequences, this in blue, in order to be expressed in plants as biofactories. But the important aspect is that after designing this peptide, we discovered that, of course, the peptide has bactericidal activity against many plant pathogenic bacteria. You can see here the cells were really in lysis, completely destroyed. There are pores in the membranes and burst of the cytoplasm. But an important aspect is that this peptide produces a strong plant response especially by overexpression of genes that are related with defense, in a similar way as flagellin, ethylene, salicylic acid, and jasmonic acid, but with differences. And this has been demonstrated in almond, in tomato, in nicotiana, in different plant species. So this is an example of how we can improve taking the idea of natural peptides, but producing engineered peptides more active. We have done proof of concept as many other companies in the way of greenhouse testing or field testing. There is only a few field tests done with peptides. In the greenhouse testing, we have work against many pathogens, for example, against fire blight, against phytoplasmas, against Xylella fastidiosa, against Xanthomonas pruni, in prunus. But in the field, we did field test against brown spot of pear with one of the peptides. This is a peptide made on 11 amino acids, all normal or regular amino acids, but also other peptides like this one, which is a defense elicitor, an endogenous peptide from Prunus dulcis, from almond. So this is an example of how we can arrive through the greenhouse testing to the field. The problem for field testing is that the cost of product is very high for doing the test, especially if you produce the peptide by chemical synthesis. And now the challenges we have with peptides, because I told you that there is a very wide world and there are thousands of compounds. So toxicity and non-target effects is an aspect which is very important. It can be tested by phytotoxicity. There are several assays that we can do, hemolytic activity. We can use even the nematode Caenorhabditis elegans as a model of toxicity. And finally, mammal toxicity with different ways. But the references, you can find that, for example, piscidin, which is a peptide that comes from a fish, is non-toxic. Surfactant have low toxicity. Surfactant is a cyclolipopeptide with biosurfactant activity. Itorin and fenticin are toxic, but less toxic, for example, than a detergent, which is very well known. But for example, peptide balls produced by trichodermas are generally toxic. So we have a wide range of compounds from natural origin, from non-toxic to very toxic. By developing analogs with synthesis, we can improve this toxicological profile. Other issue is the biodegradability, because we want that these peptides should be biodegradable, but not too much, because then if proteases hydrolyze the peptide, you lose the activity. Also, the range of action. Sometimes the range of action is too wide, so it can be also adverse effects. So these aspects should be taken at the time of regulation of the technology by the authorities. And another challenge, I'm just going to finish, is the sources of production. The sources of production are important. You can choose natural sources from any kind of living organism. The most striking and interesting is the use of microorganisms for that. And for example, one source which is probably the most important is to take advantage of in companies where there are they produce microbial biopesticides. And sometimes the culture supernatant contains great big amounts of these peptides that can be useful. But always they should do downstream processing. And downstream processings increase the cost of production, because you have to purify relatively the compound and then formulate. The chemical synthesis up to date is only interesting for research. For example, for screening libraries, for developing new compounds, but it's made by solid phase synthesis, which is very costly. And for an application in agriculture, the cost is too high. Microbial biofactories are the most interesting and probably the most used by the companies that are producing peptide insecticides at that time, because the yield is interesting, but the average yield is not too much. Also, is in the order of 1 gram per liter of peptide. Also, possibility by algal biofactories. There have been some groups developing technology for that. And finally, the plant biofactories. Plant biofactories is also another interesting approach, but in all these cases, microbial, algal, or plant biofactories, some genetical modification of the organism, microbe, algal, or plant, should be done to increase the yield of production. So production can be natural one, by chemical synthesis, by also using living platforms based on heterologous expression of the peptides. But finally, you have to purify and formulate. And the last challenge is, in one hand, is formulation. So for agriculture, any pesticide, even biopesticide, should be formulated to extend their shelf life and to increase the stability and efficacy. So formulation depends very much on the type of peptide. For example, if you have an amphipathic peptide, cationic amphipathic peptide, if in the formulation you decrease the pH or you increase the salt concentration, you change the activity and even the biological profile. So this formulation should be studied carefully. And the other challenge is how to deliver the peptide to the plant. Tentatively, you can use a conventional system, spraying, irrigation, and so on. But you are losing a lot of compound. So if you spray the trees, you lose a peptide which is costly, and this is only reasonably recommended for elicitor peptides, because in the case of elicitor peptides, generally the concentration needed is considerably lower than, for example, for the lytic peptides. And the last technology, which is under development, and there are several companies working on that. I have seen one of the company in the list that you have presented, Jennifer, before, which is in bio, which is a system of endotherapy. So endotherapy, in my view, is the best way of using peptides, especially the elicitor peptides. And this is under development. We have been collaborating with several companies in this way, but we have to do more research on that, about stability, pharmacokinetics into the plant, even residues, and a lot of work has to be done in this way. And finally, for ending my talk, if any of you are interested in to have more information or more knowledge about peptides for disease control, I invite you to read the annual reviews from myself that has been published 2 years ago, and also another chapter in a book. Or even if you go to our references, the references of my research group, you can see details on particular publications, diseases, or peptides. And that's all. Thank you for your attention, and you have here our webpage if you would like to visit us. And thank you very much for everybody to be here.
Thank you very much, Professor von Montesinos, for this very good, clear overview on peptides for disease control, and thank you for accepting to joining this webinar here. In case you have questions, I just remind you, please don't hesitate to put them in the Q&A part or chat, let's say Q&A chat that you see at the top of your screen here. And now I would like to welcome Dr. Andrea Chini, who focused his research on how plants adapt at the molecular level by employing a limited set of signaling molecules. In particular, he studies the molecular networks orchestrated by phytohormones to integrate diverse internal and environmental cues. And with this complex description, I would like to welcome Andrea Chini to present us the emerging technologies and opportunities for natural and engineered peptide. Thank you very much.
Great. Thanks a lot for introduction. Of course, the invitation to be here is a great pleasure again. And okay, we had a little issue with sharing the presentation, so I've got help from IBMA staff to switch the different slides. So if you can go to the next one, please. Great. So it was a pleasure to be here also almost 2 years ago, previous meeting, and just to remind some of the main concepts, idea that we discussed, and most of them were actually shown by Professor Montesinos. We share the presentation or the duty in presentation not to overlap, and those are things which we discuss, and I really would like to go in again. There are some very interesting, like different types of peptides and the biosynthetic mode of action. Professor Montesinos is an expert, so my focus is in the next slide, please. And basically, I would like to share what is done, most recent different strategies, especially machine learning based strategies to identify new peptides. Then to mention that natural is not synonymous safe, and you will see the reason. Going a little bit on the new genomic techniques for plant breeding for the European Union, and if we can make a analogism or parallelism with peptides, and then define or share with you some of the technology that the most recent that also open opportunity to identify or to design new peptides. Okay, next one, please. And so those are the strategies that have been conventionally classical strategy to identify natural peptides. So mass spectrometry, for sure, really getting the sample of the plants and getting out the sequence of the peptide. Most recently, ribosome profiling, which is really help to identify new peptide in plants and in nature. That's very costly, whereas mass spectrometry has different limitation, mainly the amount of samples required, and then since the very beginning, simple conservation. So to compare, once a peptide is identified, to compare if plants or different organisms can produce similar sequences, peptide with the similar sequences. And if you go to the next slide, please, we see that this idea or this technique has been really moved forward very fast in the last few years with the advent of the next generation sequences, the availability of big data, and so the sequence analysis became a completely new strategy, basically. And thanks to machine learning and artificial intelligence based methods that probably have become they have become incredibly powerful tools to predict, to design, and to search for peptides. And that's, I think, is one of the main part of this presentation. Can you go on the next slide, please? Okay, this not only my opinion, this is a special issue of Science a couple of years ago, so in 2023, and describing the new opportunity for basically based on machine learning or artificial intelligence. And one of these, in these special issues, one of the research paper or revision focus on the opportunity to identify new antimicrobial or antibiotics using peptide as a active molecules. Can you go on the next one? And one of the main scientists proposing these new ideas is Cesar de la Fuente, as you see here, and so he leads this his lab in Penn State, and I think this is basically his work is an excellent example of the power of machine learning technology in the research of peptide as with a biological activity, the identification or the design of a novel peptide. And I will share with you the results of the latest paper. If you can go on the next slide, please. And basically this has been published less than a month ago. And what they use is deep learning to search for mining archaea's genome and transcriptome, identify putative natural peptide with possible antimicrobial activity. And if you go on the next slide, please, you see that, okay, they synthesize the putative best 80 peptides, and they see that some of them are not active or little active. If you see yellow or very pale color, that means they're not active. And in the top part, I cannot use my pointer, but in the top part, they use different human pathogen, basically Escherichia coli. They use also Pseudomonas aeruginosa, for example. And the dark block you can probably see there. This means that these peptides have a very strong antimicrobial activity. As you can see, there is a mix. There are some of them. Some of these peptides are very specific against one pathogen. Some have a wide range of antimicrobial activities, some of them as known. And if you go on the next slide, since this is not really the main focus of most of the members of the IBMA, but I would like to really to show that when we move from in vitro to in vivo, in this case is a mouse model, they show that these peptides are as active as antimicrobial as some of the molecules, antibiotics that are currently used into both 2 days after infection or 4 days to different extent. So not only in vitro data, but in vivo. And now I think that's one of the many, well, one of the few examples of the power of machine learning based methods to identify peptides with a biological activity. What is more interesting to us is, can a similar approach be used in the plant protection? And that's actually what I proposed to Cesar and Marcelo a couple of years ago. We started a collaboration, and I will show you our proof of concept results in the next slide, please. Here we go. So in this case, with the same approach I showed you before, we use plant genomes available or transcriptomes available, big data, use the machine learning approaches to identify putative natural peptides. In this case, are plant-based. And in Penn State, they analyze, well, they synthesize it, they analyze the activity against the different human pathogen. They are on top, Escherichia coli, and a number of them. As you can see, blue means no activity, but among the top 80, there are many of them with other specific activity or activity against many different human pathogen. So this is in for the most interesting part for Cesar and Marcelo. And what we are doing here in Spain, where a collaborator of mine and myself, is to test the antimicrobial activity of these peptides against plant pathogen like Pseudomonas aeruginosa or Xylella fastidiosa, and the results are in the next slide, please. Great. So as you can see on the top part, the control is the growth of Pseudomonas, for instance, and the addition or the treatment in vitro with one of these peptides can prevent the growth. So antimicrobial activity. Then is quantification below, but really there is no point to go into details. We are moving toward in vivo implant analysis. So to check the activity of this peptide in planta, and we haven't results yet, but this shows that that we can use these machine learning approaches to design novel peptides based on natural genomes. So those are putative natural occurring peptides with antimicrobial activity, and same idea can be used by any of the IBMA members to look for regulator, peptide regulator of the biological activity you are most interested in. Next slide, please. So this is what just I mentioned, opportunity to identify or to define new putative natural peptides, but is also an opportunity to revise some of the peptides you're already working with, to have good results, and to look for whether these peptides or similar peptides are putatively natural. And you might have started to work with the peptides for their biological activity, but now with more and more genomic data available, big data, and these machine learning processes, it's easier to look for these peptides or related peptides in nature. And that and why it might be useful or interesting to know if a peptide is potentially natural. This is a matter that came in the OECD meeting, in peptide meeting in February, and probably I didn't explain it sufficiently well at that time. So I would like to mention, if you go on the next slide, that I think is what Professor Montesino just showed. We know that natural substances, of course, including peptide, can be harmful, can be toxic, and those are matters that are important. The fact that a peptide or a molecule is natural does not prevent toxicity, but the benefit is if we can make a parallelism or analogy with a long discussed issue in the EU, which is proposed in the next slide, please. Which is the new genome techniques. So, well, many of you are very familiar. You surely know quite well, most of you, the outcome of this very long discussion on the genome techniques is the definition of two categories of plants. If you go on the next slide, please. Category 1, which are plants equivalent to conventionally bred or naturally mutated plants, that goes through a pipeline of regulation, which is much lighter compared to the second category, where the plants are considered more complex modification and go through a more complex regulation. So the fact that the peptide can be natural, I think it might open opportunity to using this analogy, the parallelism, to propose it a similar outcome as the result of this long discussion, the new genomic techniques for plant breeding. And now, in the second half, I would like to focus on the new opportunities to identify natural peptides. Next slide, please. So this was proposed based on these evidences, the possibility to analyze environmental DNA. So basically, DNA that is extracted from, for instance, soil or environment that has been well preserved. The typical is the recently defrosted area, for instance, in Greenland or anywhere close to the North Pole or South Pole. Next slide, please. And for instance, Nature dedicated a couple of years ago a special issue on these new opportunities coming up, and there are still many limitations, but what for instance, the take-home message of this specific research, and I think is quite important, is that we, with this environmental DNA, we can actually have a look in a ecosystem that no longer exists nowadays, but we can have at least the genomic information. Genomic information means we can infer the peptides, for instance, that were produced or living organism could produce at that time. Next one, please. So that was proposed a couple of years ago with environmental DNA. That is a mix of different DNA of different living organisms, microbe, plants, animals, and you mentioned, and very short reads. But nowadays, for instance, this is an example of last week, basically. Some of you probably read it either in the news or in your favorite newspaper. So the identification of microbes from mammoth remains. And if you go on the next slide, please. Basically, what they show is they analyze the teeth, basically, of mammoths that were recently collected because of the global changing and the appearance of new remains in the last few years. And actually, they could sequence approximately 20% of the genome of a singular microbe. And this is an improvement, a very big improvement compared what I mentioned just before. So in just 2 years, without entering details, we're not interested in mammoth or microbe that live to 1 million years ago. But that show the improvement of the quality of the DNA and the opportunities to identify new, for instance, in this case, natural peptide that existed a long time ago, don't exist any longer. These microbes were adapted to different environmental conditions that are not there any longer. So they are possible source of really new peptides or molecules that we really need to look into it. Okay, and related to this research, next slide, please, is a very recent example in plant, related to plant. So can we apply this idea to plant and plant research? And in a lower timescale evolution, in this case, they focused on Nicotiana, which is the wild relative of tobacco. So in this case, the speciation of the major lineages of tobacco occurs in the last 5 to 10 million years. But on the next line, what we can do with more recent species is to identify really in vivo peptides that are natural. For instance, Lysicomicum peptide is produced in Bentamiana nowadays, and is a peptide with potential pharmaceutical application, especially as anti-hypertensive drugs, for instance. But it also show plants defense against pathogen and herbivory. And the modification of an enzyme, a biosynthetic enzyme of lysocumin, a change in that enzyme can now produce a new peptide, which is nanamine A, that is under study. So this peptide was inferred through genomic analysis, and the metabolic engineering of plant could produce it in vivo, and now the peptide is there. So what is the interest? What is the function? They don't know yet, but the idea try to exploit the evolution, plant evolution, to identify new targets, new peptide. So the plant evolved this peptide to improve fitness and to improve the survival and adaptation to the environment. Therefore, at some stage in the history, in the evolution of the plant, this peptide has an important biological activity. So that's what is an example of this new opportunity to identify and study natural peptides. Next one, please. And so what about plant paleomicrobiology? Are we there yet? This means to study the community of bacteria or microbe that, for instance, can be recovered in old seeds or plant specimen. And the truth that is still a very open, well, a very recent techniques, and we don't have a great result yet, not great results. So it's promising. There are many challenges in methodological challenges. We don't know whether it will meet the expectation or not. We will see in the next few years. And so as an alternative, or next slide, please, or hand in hand with the plant paleomicrobiology comes the analysis of herbaria. So this opportunity was proposed and emphasized in a specialization in science a couple of years ago, as I show you here, and the new sequences nowadays with the new techniques, we started to have sufficiently high quality herbaria specimens sequences. Anyway, so we're speaking about just before about paleomicrobiology, million years old specimen compared to herbaria, which are just a few hundred years old. Can we get similar, or can they compete, or can be complementary? And what is the study basically to the benefit to study herbaria? And I show you on the next slide. Basically, it's true that most of the herbaria are just a few hundred years old, but we should take into account that the specimen is quite well preserved in some cases, of course not all, and the changes that we suffer, well, the earth suffer mainly because of humankind. And of course there are some examples. I just would like to show you there the increased level of CO2, the use of herbicide that of course go in hand with the loss of habitat. And also, by our point of view, some of these herbaria can be actually witness of the crop-to-wide transformation of wide species into croppable plants. And also the great they are before the great genetic erosion that we are witnesses nowadays. So are we meeting the expectation? Can we get good results out of these techniques? And in the next slide, please, we see that they are starting some this technology, some potential study that really studies that back its potential. One example is this one, again quite recent, and where they extract DNA from herbarium, and there are 4 species which are shown in the next slide, please. And so mainly cereals, but also lettuce, and these give results which are shown in the next slide, please, which show that we can, they can identify many communities, in this case specific microbe or bacteria that lived with this plant. Some of them are pathogen, some of them are actually symbionts, and as you can see here, they're defined. So nowadays it's difficult to quantify exactly how much of the genome of these microbial can be sequenced. Probably we can estimate between 10 or 20%. So still quite little compared to the full genome that we would like to have. But then these results show that we can really have a look of genomes that co-evolved with the plants a few hundred years ago, but in a complete different environment. And having the opportunity to get, if not the complete, most of the genome of these symbionts or pathogen, and study the putative natural peptide in these samples, I think it would be a great opportunity to identify new peptides with potential different activities, and of course I think it will open a lot of possibility. And with this example, I would like to finish. Next slide. And yes, I think I just made this is the suggested time. So more than conclusion is just a few ideas for the discussion, which is summarized in the next slide, which is the last one. So I focus on the very recent advances, not because the latest results are better of the previous one. Just my idea was I wanted to highlight the astonishing ongoing technology. The advances, they are so quick, so fast, and make our knowledge so dynamic in the field, and also show how difficult, challenging it is to foresee the next step. So with that, I would like to thank you for being here, and I hope we'll be able to contribute to the discussion.
Thank you very much, Dr. Kinney. I think your presentation highlights how peptide technologies are developing fast. This is really fascinating area here, and how huge is the potential for new peptide identification, but also how challenging it is. Now we are giving the floor to the participant of our webinar. So please feel free to raise your hand or post your questions or comments in the Q&A chat. We already have one question, so we will start with a question from Patrick Cabot in the chat. Thank you so much. So that was reacting to Professor Montesino's presentation, but I'm sure we can share the appreciation to the second presentation from Dr. Kinney as well. On the challenges, how can we balance biodegradability and stability to ensure that proteins degrade into amino acids, the building block of life, while ensuring sufficient efficacy over a long enough period for the farmer? At least it is not a regulatory challenge. Endotherapy won't be the option for row crops. Montesinos, maybe?
Yes, these are two key questions. The first one is biodegradability can be managed mainly through developing analogs. And precisely one of the important aspects of our pipeline that I have shown in the first slides is that we test for protease susceptibility. So you can do this in the lab and see how resistant is the peptide to degradation. You are thinking about amino acids, but sometimes it is the final products are shorter peptide fragments, but the activity is lost. So from the regulatory view, this is good. But from the activity view or the user view, this is not good because you lose the activity. So there are other ways. For example, we use introduction of D-amino acids because, all proteins and natural proteins, most are made on L-amino acids, and proteases are specialized in hydrolyze sequences by L-amino acids. So if you introduce D-amino acid, you prevent degradation by proteases. So there are different ways, but in any case, the ways are based on developing analogs because and another important aspect is that many natural peptides are relatively resistant to proteases. For example, the cyclolipopeptides from Bacillus generally are very stable. For example, surfactant, iturin, fengicin. So the question is, as Dr. Kinney said, is look for natural compounds that fit your interest. So I'm sure that you will find anyone that is interesting. So the second about endotherapy is obviously only feasible for wooden plants, wooden trees. And for example, it is being used, tested, and developed for citrus against Huanglongbing. and for Shigella fastidiosa in olive trees, in almond, and it's a question of technology development. Now there are companies that have very good systems of endotherapy that do not require a lot of handwork. So that's the way. Evidently, you cannot use endotherapy for tomatoes or hort plants, but in my view, is the best way of administering the peptides. But for example, the plant defense elicitor peptides are effective in many cases simply by spraying the plant. Even you don't need to spray the whole plant, only one part. So it's very diverse. Yeah.
Thank you very much, Professor Montesinos. Do we have other questions in the attendance? No further question?
Jeremy Marizet added a question to my question.
Okay. Oh, yeah. Show one comment new. Okay, sorry. Thank you very much. Okay, Professor Kinney, can you comment on how proteins with identical structure and function can vary from one plant or microbe to another, i.e. alleles, and how these could be predicted and mimicked by in silico evolution rather than by directly sampling from nature?
Okay. Can you read it again? It's in the chat because I'm not sure.
It's in the
I'm not
Q&A
sure.
chat.
Okay,
Issue.
so I will read it.
Okay, great. Thank you.
Yeah. Mhm. Okay. I'm afraid I cannot get it in my chat. I don't know why, but
Would you like to
if I
read
get it
again?
right,
Read it?
yes.
Yeah.
Please.
So, can you comment on how proteins with identical structures and function can vary from one plant or microbe to another, i.e. alleles, and how this could be predicted and mimicked by in silico evolution rather than by directly sampling from nature?
Okay. Yes. In nature, there are several examples of basically mimics, especially for symbionts or micropathogen. They actually they can either in this case protein, they produce protein, encode protein or peptides which are identical or with a functionally identical to the one of the plants to try to hijack, for instance, the defense. In the case of pathogen, they want to hijack and block the defense of the plants, and they do that by producing identical or with the identical activity protein or peptide with identical activity. Again, for symbionts, they want to, well, in order to get this very close contact with the plants and to have the beneficial exchange of energy, carbon source, and different elements. Part of this communication is based on these molecules. They can be protein, they can be peptide, or some other of other nature, not pro amino acidic nature, to the communication. So we know that they exist. To predict in silico is, I think nowadays with the machine learning, train a pipeline to predict it. It will be relatively feasible. Not easy, but very relatively feasible. And of course, it depends on the. It's difficult to really to address the question. It depends on the biological activity you are interested. So normally is a pathogen, how to, as Professor Montesino just mentioned, many of these peptides don't have only a direct antimicrobial or antipathogenic activity, but they actually induce the activity plant defenses. So there are many research ongoing try to identify novel molecules with activity to promote this regulation of, for instance, plant defense, which is the field which closest to my scientific interest. But that's applied to many different biological processes.
Thank you, Dr. Kini. And we have new questions here. One from Selene Garcia Hernandez. So thank you both for the presentation. I would like to know how is the ADME, especially the absorption of peptides in plant tissues compared to other organisms such as animal or fungus?
Emilio, you're the expert.
Well, not exactly, but yes, there are some peptides that can be absorbed, but as far as we know, there is not too much studies. If you say absorption after spraying, I don't know any kind of study done, but by endotherapy, I can tell you that if you inject, for example, in an almond potted plant in the greenhouse with 30 microliters of one of the peptides I have shown, the peptide moves mainly upstream to the plant. Some part of the peptide moves to the roots, and it can stay for days or weeks, depends on the physiological state of the plant. But this is for a particular peptide. You cannot do the same for all the peptides. So we need a lot of work, a lot of research on what I call pharmacokinetics into the plant, as in humans or in animals. So in some cases, you don't need to enter to the subsystem into the plant because you can trigger defense of the plant by simply interacting with the cells of the leaf, with the epidermal or the mesophyll leaves. Cells of the leaf.
Yeah.
So it's very complex. I'm sorry, but I don't have a very simple response to your question. Yeah.
Thank you, Professor Montesinos. Another question from Christina Donat. So it seems your presentation raised some interest and a bit of debate here. The use of antibiotic peptides for plant protection is not fundamentally new. E.g., streptomycin was and is outside EU used against fire blight. So far, this was not supported in the EU. Why do you think this will be different with your products?
The difference is that if you paid attention to the streptomycin or tetracycline molecule, you see that they are complex structures. In many cases, they are relatively resistant to degradation in the field. They inhibit protein synthesis. And for example, our peptides, the mechanism is a lytic effect. So there is a killing the cell by destroying the cell membrane, for example. So there is no mechanism of resistance to that. The only way that the cell can have is to block the peptide to not arrive to the membrane, which is a way of resistance, but not comparable to the ones you said. I know very well fire blight, and I know very well how in the USA and other countries, streptomycin and tetracycline are used. These peptides are completely different from that. There are analogs also. For example, the surfactant, iturin, and fengicin from Bacillus are similar to polymyxin and gramicidin because they are cyclic peptides. So, for me, there is no difference. streptomycin is an antibiotic, and surfactant and iturin can be considered also an antibiotic in the microbiological sense. The problem is that when we say antibiotic, we think about clinical and hospital use. And this is not true. In microbiology, antibiosis is a phenomenon of inhibition even by an enzyme or by an antimicrobial. So better not confuse both. Yeah.
And one on the line that you already started to answer. Is there anything known about the resistance development of microorganisms against peptides and proteins? Therefore, yeah. Another question from Laurent Dini. A question for Dr. Montesinos. You explained the development of proteins and peptides in one of your slides. These can be diverse and have different mode of action. To regulate this technology, it could be good to classify these proteins and peptides types according to their mode of action. Which criteria to consider to properly classify them?
There are, I know, at least 7 to 10 ways of classifying peptides. And probably Dr. Chini knows also there are many ways, from the chemical point of view, from the mechanism of action, from the structural point of view, from the origin. But the mechanism of action is one way, and I think that probably if I think as a regulator, this could be an important criteria of use, the mechanism of action, because in the mechanism of action, it is implicit the range of action, the specificity of the action. And in that way, for example, you have an antifungal peptide, the range is against fungi. If you have an antibacterial, is against bacteria. So it can be a way. Yes, you're right.
Thank you. Before we head to the conclusion of this webinar, I just wanted to make sure that there are no further questions from the attendants. No? So if there are no further questions or comments, I would like to give the floor back to Jennifer for closing remarks, but I would like to give also to take this opportunity to thank you again, Emilio and Andrea, for this really insightful and passionate presentation.
Thanks.
Thank you. Jennifer,
Thank you.
The floor is yours. Thank you.
Thank you, Jeremy, and a huge thank you to our very eloquent presenters. Thank you very much, Dr. Chini, Professor Montessori, and really very fascinating presentations. I mean, we heard about the wealth of mode of actions in peptides, and then have just had a discussion about how those may be considered by regulators. How could we classify according to mode of action? But it was also an interesting discussion about the biofactories and how do we actually manage those, and the source of whether it's a microbial or a plant that we need to maybe modify in order to ensure an economic yield. So all these factors in the production of our peptides. And then when we come closer to the plant, actually the formulation or delivery of that, and how that varies by peptides. So a huge wealth of knowledge was shared, and much appreciated. Thank you very much. And then we came on to Dr. Chini's machine learning-based strategies to identify natural peptides, and what a rapidly developing field of science. Paper after paper shared with us about what and how this is now being developed, including taking ancient microbes and ancient dried plants. I had to look it up. What is a herbaria? But and I'm a native English speaker, so it's dried plants for anyone who was struggling like me. And to identify even the microbiomes associated with those. Really a fantastic area of and field of research. But you both made quite an interesting comment. I saw you nodding at each other during the questions. And that was about, you should actually look for the natural compounds that fit your strategy. And that with all these tools open to us now, that is actually much easier and much more feasible than perhaps it was in the past.
Yes.
So really, that was a huge thank you to you for really insightful presentations. A huge thank you to our at one stage 111 participants. We've got a few have left now, but really a big thank you to all our participants for the great questions and for listening and for showing your appreciation. So with that, I would like to just make one further comment for the IBMA members, and that is that you will have noticed that there is a market survey open for our members, and it is very, very important that we have every company complete this. And the reason is because this evidence, this market size information is being asked for by the Commission and by parliamentarians as we enter these potential simplification procedures and changes to 1107 for biocontrol. So please do the market survey, super important. And with that, I want to thank everybody, thank especially our speakers, thank Jeremy and Bridget behind the scenes for all your help. Thank you very much and have a good afternoon.
Thank you very much.
Thanks a lot. Great,
Bye-bye.
thanks.
Thanks.